Today I'm going to be talking about glycolysis. This is the first step in cellular respiration. During glycolysis, glucose it also requires 2 molecules of NAD+, which lead to make 2 molecules of NADH, and also for this process, 1 molecule of glucose, which creates 2 molecules of pyruvate. And that is glycolysis.
Okay, so you have three carbon glucose molecule with a phosphate group attached. One more phosphate group is attached to each end of each of these. NAD plus is converted to NADH plus H plus. 2-ADP is converted to 2-ATP for each of these molecules. So, and then in the end you have 2 molecules of pyruvate. And in the end, you have 2 ATP molecules on this for this molecule and 2 ATP molecules for this molecule. So you get 4 ATP molecules in total. And you lost 2 ATP molecules from here, which means that you have a net of 2 ATP molecules, a net of 2 pyruvate molecules. So this is the process of glycolysis illustrated in one handy diagram. Yeah. So first, with glycolysis, you start with 1 glucose molecule with 6 carbons.
Good boy.
So then you dephosphorylate ATP, 2 ATP into 2 ADP. Phosphate groups are added to each end of the glucose molecule. The glucose molecule is split into 2 molecules, each with 1 phosphate group. 1 phosphate group is added after
Forget the key.
What?
Okay, so I'm going to be talking about the Krebs cycle. The first thing to know about the Krebs cycle is that it occurs in the mitochondrial matrix. The process of the Krebs cycle is aerobic, which means that it does not require oxygen. The input for this process is 1 molecule of pyruvate. However, this process is actually, it goes through twice, so you have 2 molecules of pyruvate. But for this, I'm just going to write the output of 1 molecule. So 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of ATP, and 1 molecule of carbon dioxide. So because this process occurs twice, you're going to have to double each of these numbers. So the total output is going to be 6 molecules of NADH, 2 molecules of FADH2, 2 molecules of ATP, and 2 molecules of carbon dioxide. Okay, so I'm going to be introducing the anatomy of the mitochondria and also some of the processes that go on inside of it. So first of all, the mitochondria is an organelle, so it can be found inside a cell. So the first thing you have to know about the mitochondria is that it has its own DNA. And this line right here, this is the outer membrane. This space right here is called the inner membrane space. And this second line right here is the inner membrane. And the last part of the mitochondria is this space inside here, and this is what's known as the mitochondrial matrix. Okay, so those are the four basic parts to this organelle, and now I'm going to talk about the processes that go on inside it. So most importantly, in your mitochondrial matrix, you have the Krebs cycle, and that occurs in there. And also, through the inner membrane, protons are pumped out in this direction towards the inner membrane space via a proton pump. These protons then move back into the matrix through ATP synthase. And so that would be from the inner membrane space into the mitochondrial matrix.
Okay, I'm going to talk about photosynthesis. Photosynthesis is a process by which light energy is converted to chemical bond energy. There are two main types of photosynthesis, the light-independent reaction and the light-dependent reaction. And obviously, the light-dependent reaction requires light, and the light-independent reaction consists of Calvin cycle, which produces sugar. And because the Calvin cycle is fueled by ATP from the light-dependent reaction, so both reaction has to occur only when light is present. And photosynthesis happens in a chloroplast. Chloroplast contains grana, and it is where light-dependent reaction occurs. And since the light-dependent reaction is fueled by light, it is also called photophosphorylation. The purpose of photophosphorylation is to convert light energy to ATP and NADPH to be used in the Calvin cycle as two parts, the cyclic and the non-cyclic part. The cyclic part only produces ATP, and the non-cyclic part produces ATP and NADPH. And the Calvin cycle needs large amount of energy, so it had done so by phosphorylating ADP into ATP and in a series of electron transport chain. And during the non-cyclic full phosphorylation, electrons enters two electron transport chain, and ATP and NADPH are formed. NADPH from the light reaction are used by the Calvin cycle to make glucose. So you take CO2 plus NADPH plus ATP, and it converts to glucose, which is C6H12O6, and one glucose is made per every 6 Kelvin cycles. And that's it.
Alright, we're going to talk about oxidative phosphorylation, and that can be broken up into the electron transport chain and chemiosmosis. So in the electron transport chain, protons, or the H plus ions from NADH and FADH2 are pumped from mitochondrial matrix to inner membrane space. That creates a gradient, and then the H plus ions want to move back in. And then electrons move through the membrane via electron protein carriers. And then this also exists in the chloroplast of plants. So that was electron transport chain. Now we're going to do chemiosmosis. Alright, for this one, protons travel through an ATP synthase down the gradient into the mitochondrial matrix. This provides the energy to phosphorylate ADP into ATP, and then it creates 32 net ADP. Yeah. Hey, hi guys. So we're going to review Gibbs free energy. You guys might see this on the Oxford Pews, or maybe the multiple choice, or the math grading questions. So this is Gibbs free energy equation. So it's basically the change in Gibbs free energy, or free energy, equals the change in enthalpy minus the change in temperature times the change in entropy. So again, as you can see here, G equals free energy, H equals enthalpy, T equals temperature, and S equals entropy. So basically, you just need to know these four things. If your reaction is exothermic, then the change in enthalpy is less than 0, or negative basically. If it's endothermic, then the change in enthalpy is greater than 0, or positive. If the reaction is exergonic, then the change in the free energy is less than 0, negative. If it's endergonic, then the change in free energy is greater than 0, positive. And remember that exothermic is you're releasing energy in the reaction. Endothermic is that the reaction is absorbing energy. Exergonic and endergonic relate to the decrease or increase in free energy respectively.